discussing some of the topics related to scientific writing. Apart from those things we have already discussed in class, some of the topics we'll be revising and revisiting so for better understanding. So when we are doing scientific writing, some rules and regulations probably we will discuss. Of course, this is very much theoretical, but you need to understand in perspective of different component. So when you are writing a scientific article, the material and method section is really very important, and we need to know what are the component it should have, what should be the property. Many things we already know. So essentially, the material and method section, the purpose is the experimental work whatever we have carried out, how to represent it in a connected and logical manner. That is the objective. So method section we should write in such a way that it will have enough information so that anyone, the a new researcher can repeat the experiments in the same way. So reproducibility of the experiments so that we can repeat it is very important. So your material method section must be sufficiently detailed to repeat whatever the study is going to tell by another person. It is really very important when you are writing any material and method. It should be clear from the method section how all the data in the result section were obtained. So whenever someone is reading the results part, he or she must be able to know how the result data came out. So it should be really the building block of the whole study, how you are doing the experiments, how you are going to take the samples, how you are analyzing. Those aspects must be there in sufficient details. But sufficient detail doesn't mean that each and every step should be written there. If some steps which is very common and regularly used by scientists, repeatedly used, we can just put the reference of that, and how we are modifying or changing those protocols we only need to tell those basic steps. So it is acceptable. You need not tell every detail if it is really a known and widely used method known by scientists or researcher in a global perspective. So those things you should take care. The study system should be clearly described. Researcher need to specify how many number of samples you are taking, how you are going to take, when you are taking, where the subjects were recruited, from where you got the things. For example, I am doing some soil study, then I need to tell of course the detail from where I get the soil, how much soil I collected, with the soil what I did, make it dry, make it sieving, I am keeping it, then what I am doing, keeping it in a freeze, keeping it in a room ambient atmosphere, then what you are doing. So for example, I am taking some amount of soil, mixing with water, in which volume of water I am mixing. So all the details, technical details, which is really important, you need to mention, you need to write those things which are really important, which can be included in the study. On what basis I chose those particularly soil or that sample need to be included in this design when you are talking about material and method. In most cases, experiments should include appropriate control. So most of the time, if I am doing some extraction, if I am going to do a study which is going to show you cause and effect aspect, then you must have something control. It can be positive control, it can be negative control. uh, in the same manner, for example, you want to study biodegradation of one compound, or maybe you have identified one microorganism which can degrade some of the plastics. So it is fine, we want to evaluate that aspect. So what should be the positive control? So that means those compounds, maybe I am using the microorganism, but I am giving those monomer of the plastic degradation product. So we are giving the food which is really produced by plastic degradation. So this is the scenario where positive growth, microorganism will definitely grow, Negative scenario, I'm giving plastic but I'm not taking any microorganism. That means obviously there should be no degradation at all. So I can see one scenario where there is no result possible, another scenario where definitely result is possible. So we can actually measure, we can scale our outcome. So we need to have these kinds of control in all our study. We need to write it properly in our experiments. We should need to mention it, that what is the positive control, what is the negative control in our writing. Outcome of the study should be defined. So in each experiment, what we are going to do, we are going to analyze this analysis of polycyclic aromatic hydrocarbons, analysis of heavy metal. What is the each step, what is the objective? Extraction of soil, maybe for example I'm giving a subheading in material method. It means that we're going to do the extracting study using different variables. So we need to tell what you're going to do, what is the outcome you're going to measure, and it should be validated in the study. So material method section should be very clear. It should include whatever is sufficiently justifying the experiments. The methodology should be very clear so that one can repeat. It should be valid enough to tell if there is any statistical design is included in the study. You need to tell all those aspects together. Still, yeah, that I'm going to discuss now. The method used to analyze data must be statistically sound. So whether I am taking a single sample or I am taking a triplicate and I'm doing the analysis based on the triplicate analysis. So I'm going to compare two groups. So maybe I can use one ANOVA. I'm comparing cause and effect analysis. I'm treating with something without, for example, I'm treating some of the cell with some bioactive compound, another one is without bioactive compound, and then I am studying how much glucose uptake. So two scenario I am taking. So of course here cause effect is there. So one group of microorganism, first I am incubating in some antibiotic compound, then I am taking those microorganism and putting in biodegradation study. That means the same group, same cell I am treating somewhere, then I am taking it somewhere else. That means it is a cause and effect analysis, and in that kind of study we need to have what kind of statistical analysis we should do. We need to adapt appropriate study. For example, in this case I will use a paired t-test possibly. So you need to understand what kind of study you are going to use. So if I'm doing some qualitative study, we need to tell what is the appropriate study questions over there, what is the qualitative research method involved there. Maybe in other department or other schools, the methodology may be different from those who are working in engineering or science discipline, but still it is very much important to know and understand this study question. If the author has used some technique from published study, then you should include a citation and a summary of the procedure. I already told you need not repeat the whole things. Only what is the summary of the procedure and which is appropriate, whether the method is appropriate or not, you need to tell and justify. All material instruments should be identified. What are the supplier, from which company, which test was conducted, model number, all those things should also include in your material and instrument section. The method section should not have information that belongs to other section. Of course, you cannot put any result over there. It is not justified. You cannot introduce why you are analyzing this instrument. All those aspects you should not have there. So neither introduction part, neither result part should be there in your material and method. You should be very much clear on that aspect. Now in next two slides we will be discussing about results and using figure or even table. How should we write, how should we take care about result and probably figure, table, those aspects. Result section including any figure and table are some of the most important part of the manuscript, because when I am getting one new manuscript to review or maybe to read as a new scientific paper, very quickly what I normally check is the abstract part, and then I normally go into result section where I look for table figure which give me quick idea what are the things are there, what kind of study is involved there. So that is very important. Of course I check result and discussion other component also, maybe some statistical analysis, some segment of material and method we look, but result and discussion gives us very clarity what is the breadth of the work, what extent the work has been done. You can find out just checking the table and figures in a manuscript, which is mostly coming out of results section. For figure, you must check, one should check whether the plotted parameters are clearly defined. Sometime we will have a figure without knowing that what is the x-axis, what is y-axis, what is the unit of those axes. Everything should be very clearly and precisely given. If I am having many, maybe more than one line drawings are there, more than one bar is there, so I need to use the legends for which legends is going to say for what part. So different color, maybe in legend entry we should tell what are the component very clearly mentioned inside the figure itself. You need not take it outside. But figure text, if I am going to discuss what is the figure, you must not write inside the figure, it should be outside the figure part. So those aspects you need to take. What is the figure should be there, figure and table should include such. Also it should also include like standard error, confidence interval, or error bar in a figure should be there. You must also mention how many, maybe in figure caption you can mention, or table also you can mention as footnote how many sample was there, so number of sample, and you are using standard error or maybe standard deviation. Those aspects you should clarify. Table headings and figure legends should be detailed enough that reader can understand the data without reading the main text. So figure and table should ideally be standalone. I need not go to the manuscript to understand what the figure says. So that is ideally required, but it is not really possible every time that all the data, all the figure can be understood without referring the manuscript at all. It is not like that. But in most of the time we are expecting you should have so much detail possible, as much as possible you should write. In the legend text or figure heading, including figure, you can have footnotes to tell all those information. So they should be standalone ideally. Data should not repeat in figure, table and main text. So for example, I am doing some study where we are growing some microbial culture using some nutrients. So there is two nutrients. For example, I am using glucose and fructose, and I am growing for 7 days microorganism, and after 7 days I am going to check what is the total biomass. So what is the result for us? Only two result, biomass after 7 days. So for example, 1.5 gram with glucose and 1.7 gram with fructose, just two values. Do you think we should make a graph on that, maybe bar chart using simply this 2 bar? Is it important? No. This is so nominal, this information is so nominal here that it doesn't require to make a figure, because it is simply one line I can write. But if you are having more complex information, for example, each day you are measuring glucose, you are measuring biomass concentration, how is it changing, so that means it is no longer a single values, it is a trend. So it require a figure, definitely not a table. Table will not look a good one. So figure gives more easy understanding for any information rather than even table. So you need to judge, the author need to judge which kind of information I will put in figure, which kind of information I should put in table, and we must be very sure that if I am writing in figure and table and text, everything we should not write the same thing. In text part we will only highlight the important things. We cannot write the whole figure in a textual term, not possible. So text should only point out what is the key finding or key trend, increasing or decreasing, like that. But figure will give you the scientific detail if you want to know what really happens. So that part is supported by figure. Similarly, table also, I can have a big table, but definitely all points I need not refer in text, only the key points I should highlight. but for completeness I must have the entire table. Similarly for figure, and if it is very small amount of information, we can either remove those figures not necessary, I can put in text, or I can add up with another figure or table if possible. So you must be judicious whether I should put a figure, I should put a table. Is it required to understand? Is it essentially required? Those queries and those questions must come into your mind. And if the result is not really central to your study aim, sometimes we do some supportive study which is not really important. So if it is not really so important core to your research, or maybe the objective of the article, then it is acceptable that you only give the summary without giving any figure and table. It is fine. Simply summary data is sufficient, and you can simply say like data not shown. It is not necessarily required to have all the data, but if it is very important data and if you are not showing, and too many times you are repeatedly telling data not shown, data not shown, it is really not acceptable. It is makes irritation to the readers, as well as reviewer will find this is a big problem in your manuscript. So you should not have those kinds of pitfall in your writing. Interesting data, very interesting maybe, but here it is not really in the line of your main study. It is not main conclusion. Whatever maybe it is interesting, if it is not falling in your main domain of your work, what you are going to present in that particular paper, then you should put in a supplementary material. Supplementary material is really very important aspect to showcase some extra data which may be important, which may be useful for advanced reader. You can put in supplementary material. Now, is the data presented in most appropriate manner? For example, a table is being used where a graph would be increased clarity. You see, always a figure is better choice when you want to show the important things, important highlights. But table may be also useful when, for example, there are three values which are not so distinctively noted in a figure. For example, I am showing some microbial growth, 3 microorganisms, and I am finding value of 200.5, maybe 202.5, 207.5. So if I make a plot, essentially this is so big and the gap is so small, it will not be at all visible. So even if the figure is correct, technically correct, but it does not show the differences. And if my intention is to show the difference, then I will try to put a table. So you need to judge what you want to showcase, what you want to project. Is the figure, or is it sufficient to show in a figure? Is it sufficient to show in a table? Is the data too complex to show in a table? Or understanding is not very clear. So you must have a judicious approach, or rather will say you must have very good thinking process, how you can represent the same data in a different way, so that understanding makes more clarity, more easy to compare different results, different scale, normalized. Those aspects should be a paramount important when you are making figure or table. And result section should only simply state what the result were, not what they mean. So if I am having a separate result and discussion section, you must be very careful that the result section should only highlight the results, the important things. Why I got the result? Is this result important? These queries need not to be answered here. That can be discussed. Why this kind of result? What is your expectation? Why you got this? These parts are from discussion. It is not from result part. But if your article or if the journal you are publishing has a combined result and discussion things, then you may connect these dots together and you can present in a better way. So essentially these 4 slides I wanted to make you sure that you understand what is the key points in a material method. When I am writing a result and discussion, what should be there, what should not be there, how should you organize those aspects we try to make you understand, so that when you are adapting the scientific writing, you can keep those aspects in your mind. Rather, you will be habituated, you might be practicing those aspects in real life applications.